Thank you very much. It is such a pleasure to be here. When I walked in the door yesterday morning, Esper said, "Welcome home, Tom," and I'm happy to be back home. So I'd like to talk to you about some of the work that my lab has been doing to try to understand transmission. We've really made some great progress recently, and I want to share that with you. And then, if we have some time, I want to talk to you a little bit about another research life of mine where we've made some significant progress. But if we don't have time, I will stop before then. So basically, what I really want to talk about and focus on today is just this our quest to identify the first cells that get infected after mucosal exposure in, for instance, in a rhesus macaque model. And then one of the things I tried to do and finished about 30 seconds ago was to add Portuguese, thanks to Google, into all my slides. So I'm hoping that's helpful, and at least had a little bit of checking to make sure that the translation was okay. So I'm hoping that's going to help you along the way, and then for my other colleagues here, I kept plenty of English because I don't want them to be feeling left out. So anyway, the goal was to identify the first cells that get infected, and the strategy was to use a vector. And the reason we chose a vector is that it can't replicate. So any events you see that are infection or transduction have to have come from the inoculum, the original inoculum. So the downside of this is that this isn't a real virus, but we'll address that a little bit later. So finding these very rare initiation events is a little bit like looking for a needle in a haystack. And so the strategy was to use luciferase, and we often see these kinds of studies in mice where you can look for the photons being produced. So the luciferase will make the needle glow. So then you can reach into the pile of hay and hold it up and then ask, do I have the handful of hay I have, is the glowing still there? So if the answer is yes, then you can kind of sort through that. And then we use a fluorescent protein to actually see the cells themselves, the needle in the haystack. And we can validate the fluorescent protein by doing something called spectral imaging. So each fluorescent protein, each fluor, has a very specific spectrum, and if you see that spectrum, you can begin to conclude that you found this very, very rare event. And when you're looking for very, very rare events, you really want to be sure you found what you're looking for, because you'll find things that seem to be what you're looking for, but a lot of times they're not exactly what you're looking for. So what we ended up here is a new way to look at the early first transmission events. And so these are all transductions, not infections. We're using a high titer of this vector, and it's pseudotyped with an envelope called a JRFL. That probably, looking back, wasn't the best choice, but that was the choice we made because it worked the best. And when you're trying to make a system like this, you need a signal to optimize it. If you start right away saying it has to be perfect, I need to do this, this, and this, and you don't get a signal, you have nothing to work with. So we chose the circumstances that worked the best, and then we went from there. We're doing vaginal and rectal challenges. We do a biopsy before exposure. And this is basically making a break in the epithelium. So we're kind of giving the virus a chance to get into the tissue and to give us a signal. And then all of these macaque studies are done with my good friend and colleague, Ron Deasy, down at the Tulane National Primate Research Center near New Orleans. And then they pack up the tissue on ice and send it to Chicago for us to analyze. So this is an example of what the data looks like. So we take the tissues and we soak them in luciferin, we inject them with luciferin, we immerse them in luciferin because we don't have the nice circulation, say, in a mouse to push the drug everywhere. And so this is before we look and collect photons. And here you can see one of these biopsy sites. Now we start to collect the photons, and you can see in this experiment the biopsy site is producing a lot of photons. interestingly, these wounds, these simulated wounds, biopsies don't always get that infected, and it's probably because of you can have blood clots that form, et cetera, and those can really provide protection. So what we're going to do here is take this piece of tissue that's producing some photons, cut it into some smaller pieces, repeat the experiment. We have this piece of tissue here that has lots of photons being produced. We cut that down even smaller, and we end up with this, actually 2 pieces of tissues we could look at, but we're going to look at this one right here. And this is now about 2 by 2 millimeters, which is very small. So we've gone from a fairly large piece of tissue down to a little piece of tissue that we can look at in great detail. So, so the key, as I mentioned, was to do the validation. So we validate in 2 ways. One is we stain the tissue for luciferin, or for luciferase, because the vector expresses both. So you can see here that, that this is a luciferase positive cell. And then we do the spectral imaging, and in this case, it's the cherry fluorescent protein, and it has a very specific spectra with a peak at 610 nanometers of light. This was all published a while ago, and now that we have these cells, we can stain for things like CD4, CD3, CD68, and see if they're macrophages, are they T cells, et cetera. And so one of the biggest surprises that came out of this first study was that we found the virus could make it all the way to the ovaries in the female reproductive tract. And I don't have time to belabor this, but we keep seeing this again and again. And I think that it's an important consideration for a number of reasons. But since this is published, although I needed to set things up, this is just the primary data set from that paper, this paper here. And so when we look for the fluorescence, or sorry, the luminescence, the photons, we see that most of the infection is happening in the vaginal vault. The second hot spot is the ovary, and we get a little bit of infection in draining lymph nodes and in the endocervix. We really like to validate it by finding the specific cells, and that can be challenging because some pieces of the tissue are so fluorescent by themselves, very autofluorescent, you can't see those cells. So we basically, there's a few events like in this animal, we couldn't find the cells in the lymph node, but really, once again, the primary sites are the vagina and ectocervix, the vaginal vaults, and the ovaries. And then all of the cells were CD4 positive. That's good because HIV needs CD4 to infect them. And then about 70% of the cells were T cells, even though this JRFL envelope is a very broad tropism and is known to be able to efficiently infect macrophages, for instance. So the conclusions here is that it's possible to find the first cells that are infected with this dual reporter vector, that these transductions can happen throughout the entire reproductive tract, and that with the JRFL macrophage tropic envelope, still about 70-75% of the cells are T cells. So I'm gonna diverge here a little bit and talk to about something a concept that's emerging and I think going to become very important. And so it's the idea of exposure, infection, and protection, right? And so typically we defined infection as virus in the blood, systemic infection. We don't see any virus in the blood. That means no cells ever got infected, and we had sterilizing protection. And that's what's here. But the question is this true? It could be. I don't know. Maybe not. And I think that especially now as PrEP is being rolled out and we're doing other sorts of interventions, I think that the picture is changing actually quite a bit. And a nice example was published last year using a injectable formulation of cabotegravir. And so this is a formulation, and this shows the drug levels. So the drug levels go up, and then they will dip down as the nanoparticles are releasing the drug. And then you can give them a second injection, and then slowly over time, depending on the animal, the drug will disappear over time. So in this experiment, they did high-dose intravaginal challenges, basically at 1 week, at these three time points, 1 week, 4 weeks, and 8 weeks. And what's interesting here is that typically you see the virus come up in the blood systemically within a week or so. However, especially notice this animal here. This animal, the virus showed up 7 weeks after the last exposure. So basically, the only, I think, interpretation of this, or anyway, so either the virus sat around for 7 weeks waiting until the drug went away and then infected a cell and spread systemically, or somewhere in the system there was an infected cells or a little focus of infected cells that couldn't get going, but until the drug levels, because if you notice here, the drug levels in the red animal then dipped right about here below the levels with this dotted line for protection, and boom, the virus came up. So in the literature, and I don't have time to go over all of these, but there are multiple examples now in humanized mice and in other conditions where the virus comes up. Dan Baruch, at least at the Gates meeting last December, presented evidence that even in the presence of broadly neutralizing antibodies, they can see some evidence of early replication that gets snuffed out before it becomes systemic. And it's a little bit like some of the what Barney talked about earlier today. So the same could be true for microbicides, which is essentially microbicides is no longer a cool word. It was for many years, so now we call it topical PrEP. And topical PrEP was kind of being riding off into the sunset, and then recently there was I think I spelled that wrong, but a dapivirine intravaginal ring that this looks pretty bad, 27% protection, but it turned out that there were problems with compliance. The women weren't necessarily using the ring, et cetera. So depending on how you look at the data, there's some promise there. But even under the ideal circumstances where the ring was used properly and drug was detected at every assay, it was about 60% protection. And the question is, can you ever have something like an IVR or a microbicide that might protect for 100%? So I'm working with a group that includes Patrick Kaiser, who is the inventor of some next generation rings, and Betsy Harold and others. And and then using a derivative of tenofovir called TDF. And the reason we were curious about this is this idea that we could infect cells in the ovary, right? So the ring is in the vaginal vault. It's releasing drug in the vaginal vault. Is there enough drug in the system to make it to the ovaries to provide protection? And in a PNAS paper, this group in collaboration with CDC basically could show if they gave multiple low doses, they could get all of the animals infected over so many doses. But if they used this TDF intravaginal ring, they saw no infection over time. So this is one of these ideas that it worked perfectly and there was great protection. But if we then take our vector system and ask, okay, so if we give the animals these intravaginal rings, we let them go for a month, and then we inoculate with this vector, what happens? Well, what happens is we very rarely see infection in the vaginal vault, where we would see a lot of infection. That's protected, but we still can detect infection in the ovaries. What's neat is we can take the same piece of tissue and extract pieces of tissue and determine very carefully the drug levels that are present. And what you can see here is near the cervix, where the ring is located, you have fairly high levels of drugs. But as you go into the upper reproductive tract, there are very low levels of drugs. So this is consistent with the idea that you can have low levels of infection locally. These kinds of drugs are TDF is unique. Tenofovir, it's a nucleotide analog, so it has to get in the cell, be phosphorylated, and accumulate. And one idea is, for instance, in the tenofovir gel study that was done in Africa, Caprisa 004, not all the women were protected, and if they had evidence of inflammation in their female reproductive tract, in their vaginal vault, there was a higher level of infection, whether or not you use the gel properly. And so the idea is that if you have cells migrating in and out, or one model is the cell comes in and it takes a few days to accumulate enough drug to be protected, and if you have a lot of flux of cells moving in and out, you're going to have cells there that aren't protected by the drug because they haven't been able to accumulate enough of it. So, but I think as PrEP is being rolled out and considered, I think there's going to be more and more examples of cases where maybe when someone stops PrEP or is using it intermittently, there could be infections there and cases of this. And maybe this could be solved by, of course, systemic PrEP, taking a pill or using some sort of a sustained release kind of a protocol. Maybe also watching very carefully when someone stops using PrEP to make sure that this doesn't show up. So beyond using this vector to study, to basically do in vivo PK/PD, because you ask where the drug is, how much is there, and did it protect or not, we've also begun to use this to identify the sites where real SIV infection is taking place. And this, I didn't have time to put it on my slides, but this paper came out this week in Cell Host and Microbe, and I'm glad to share the link. They're kind enough to give you a link that for about a month anybody can download the paper before you have to be a subscriber or have an institutional subscription. But essentially, the idea was that this vector kind of is mapping sites of susceptibility throughout the female reproductive tract. And you know, this vector has a lot of characteristics of the real virus, but it's not the real virus. So what we decided to do is just mix real SIV MAC239 with the vector and see if the vector, the photons being produced, could tell us where the real infection was. And the answer is it could. So we started just by doing a simple PCR analysis. And what you can see here is this is PCR for the cherry fluorescent protein for the vector. This is PCR for viral proteins. So this is SIV MAC. And everywhere you saw the vector present here, you also saw the virus was present. So, and then this just gives you a map of the 5 animals in this study, which shows that with real SIV like the vector, you can get infection throughout the female reproductive tract. In this study, we found infection in the labia, we found infection in the uterus. So basically, the whole reproductive tract is potentially susceptible. So if we go into these pieces of tissue, we can find these little foci of infection, and this is 48 hours post-exposure. So the animal is exposed, 48 hours, there's a necropsy. We look for the photons, and then we take sections and going looking for the virus. And this just gives you an idea. The blue here is infected cells, and that corresponds to each one of these boxes here. So we find, most of the time at this 48 hours, we find a foci of infection. This is a very thin section, so this is sort of a 2-dimensional look at a 3-dimensional event. And so these foci at 48 hours consist of 20 to 40 cells. And we have some work to do. We don't know if this was one virus that spread, or because it's a region of susceptibility and we're using a fairly high titer, this could be initiated by more than one viral particle. If this is one viral particle, I think everything is happening faster in vivo than we realize. So again, as I mentioned earlier, when you do this kind of an experiment and you find these rare things, you really want to be able to argue that this is a real event. These are real SIV infected cells. So what we decided to do was look at some of the things that happen to an SIV infected cell, and I'm not going to go into all of them. You can look at the paper, but one of the things that happens is CD4, through interactions with NEF and envelope, becomes internalized and then degraded. So if you look here, this red cell, red is envelope and blue is internally is GAG. And you can see, in these cells around it, you have the CD4 on the cell surface, as it should be. But in this cell, the CD4 has been internalized. So when we look at these infected cells, we see various levels of CD4, from levels that are pretty much normal down to levels where we don't really see any at all. So if we quantify this, we looked at it in two ways over the area of the cell. One was the overall CD4 fluorescence, and then secondly was something that we call the radius of gyration, which is just the area that the CD4 is located. So if you remember in that previous image, as the area got smaller, the radius of gyration is getting smaller. So in the blue dots here, this is SIV minus cells from the same animals, and you can see that they have a bit of a spectrum of the fluorescence, but they all have a fairly large radius of gyration. However, in the SIV positive cells, we see two things happening. One is that there's an overall decrease in fluorescence, and secondly, there's a shift in the radius of gyration down to those cells where the CD4 is in a very small area. We can look at the same cells in a different way, and that's for the expression levels of envelope. The assumption is that the longer the cell's been infected, the more envelope it's going to have expressed. And what we can see is that the more envelope there is, the more shifted the radius of gyration of the CD4. So again, this isn't the perfect experiment, but it's consistent that the longer the cell's infected, the less CD4 there will be, and the more it's going to be internalized. So one of the things we wanted to do fairly quickly was to get a sense of what kind of cells were being infected. And so I'm glad to discuss all these markers with you later, but the one we're interested in is CCR6. And CCR6 is a marker of Th17 cells, and we've heard about those a couple times here. And inspired by the reviewers for the most part, there are quite a few experiments presented in this manuscript where we really show that CCR6 is a great marker for Th17 cells. And a lot of that, I think, was a very good thing to drill deeper in, because this is sort of better characterized in humans than it is in macaques. So we kind of had to go and show in the macaques that it was behaving the same. But anyway, the outcome of this is that there is a big preference at 48 hours post-exposure for the infection of these Th17 cells. So 75% of the cells that are infected are T cells. Of those, 84% of them are these Th17s. Of the other 25%, most of the remaining infected cells are immature dendritic cells. So we haven't drilled as deeply on that. And so what are Th17 cells? They've been mentioned a few times here. Basically, they are at epithelial barriers, and they're kind of monitoring things, and they can tell the epithelium you're not working well here. There's a problem. Could you make it stronger? And this can have different effects. And in fact, plaque psoriasis is caused by overactivity of these Th17s, which then causes the keratinocytes to overproliferate, and that overproliferation can be as problematic as underproliferation. So relating to HIV, it's been known for a long time that these Th17 cells are depleted very early in infection. Here's some of for both HIV and SIV. And then if you take the cells out of culture, they are also preferentially infected. And then and it doesn't seem to be derivative of the receptors on the cells, because you can pseudotype these viruses with VSV-G or something so it can get into most any cells, and you still primarily infect these Th17 cells. So these Th17 cells are an optimal environment for viral replication, and I think the virus really likes to interact with these cells and deplete them. And so, we have more experiments to do, but, why would these cells be the preferred targets of infection? And then I think this is consistent with the immature dendritic cells, too. Both of those cells are there at the epithelium, and they have a job to do. They have to be awake, metabolically active, sort of monitoring what's going on. A lot of the other T cells, they're kind of these effector memory cells, other memory cells, and they're kind of, they have a very important job to do, but they may have to wait a long time to bump into their antigen and start to participate in immune response. So they're almost in this state of suspended animation, and that's actually, I think, not a good environment for viral replication. And so that's why A lot of these cells, when people do things with blood cells, for instance, they activate the cells and then they become pretty good targets for the virus. But if you don't activate them, they aren't. So we have a lot more work to do, but these cells are clearly a preference of the virus at these earliest time points, and now we need to try to go forward and understand the mechanism of it. So I mentioned earlier that we do also been looking at rectal transmission. And rectal transmission has gave us a number of challenges, and the biggest challenge, and there's sort of no nice way to say this, but fecal matter is very autofluorescent. So you have all of this fluorescence, and you're trying to look at fluorescence, and sometimes, we have a saying in the U.S. when referring to that part of your anatomy, it's where the sun don't shine. Well, that microbiome that Alan was talking about, sometimes it produces photons, and we'll get an animal, and it's just, either vaginally or rectally, and it's just all lit up, and we just kind of put our heads down and walk away because we've just spent a lot of money on a macaque that we can't get any data out of. But so we do biopsies, and we've also found that there's places where the virus gets through, not biopsies, and I didn't mention this earlier, but one limitation of this system is we can't detect photons from one cell. So we need a few cells that are transduced close by each other to make enough photons to detect. And so in the rectal compartment, if we do PCR in this tissue, we can find places all around where there's one virus here, one virion there, but we don't get the photons. So whereas in the female reproductive tract, it seems like the barrier works very well, except for little places where the barrier is broken down. And where the barrier breaks down, it's easier for multiple viruses to get through and to produce the photons. So the rectal transmission work is ongoing, but I'll just give you a little bit of a taste of what we're seeing. And so that was probably the wrong term too. Anyway, so we switched to a different fluorescent protein. So this is IRFP 670. It's more red shifted, and sort of natural fluorescence it tends to be more in the blues and the greens, and maybe starting to get red, but there's not much in the far red. And then we stain the cell for luciferase, as I said earlier. So it's luciferase positive and the fluorescent protein positive. And then we can stain for CD4, so you can see that this is probably a T cell that's been infected by the vector. Here is looking at another marker, CD3. And again, we see this is a CD3 positive cell, probably a T cell. This is now looking at the CCR6, which is that marker for, actually, it's also a marker on the immature dendritic cells, but this appears to be, because it's CD3 positive and CCR6 positive, this would be a Th17 cell. And so this work is ongoing, but really it looks like, again, the main target is T cells. More than half of them are T cells. And so far in the rectal compartment, the skewing towards the Th17 is even greater than in the female reproductive tract. We're pretty close to 100% right now. So that work is ongoing. So conclusions 2 is the lich. We call it lich because it's luciferase iris cherry reporter. Can be used to study in vivo PK and PD and identify sites of susceptibility to SIV infection. We need to be cautious of occult infection with systemic and topical PrEP. And then the theme is emerging that these Th17 cells are the preferred early targets of mucosal transmission, along with these immature dendritic cells, at least in the female reproductive tract and the rectal compartment. We're also looking at some other modes of transmission, and we're a ways behind there. So I have a little bit of time to go into to something else. And so to my English-speaking friends, this is, and now for something completely different. And so what I want to talk about is the viral life cycle. So this is actually, we didn't start doing this imaging and studying sort of the cell biology of HIV, looking at whole animals and tissues, but we started by looking just at individual cells. And so what we really wanted to be able to do was to use fluorescent methods to follow the virus as it fuses and enters, and I'll show you in a second, but we were the first ones to show that the virus uses the microtubule network to move around within cells. reverse transcribing along the way to ultimately enter through a nuclear pore, deliver the genome, and integrate into the DNA of the virus. And the thing that I want to focus on right now is this question of this capsid structure here. So inside of the virion, I should probably have a nice EM picture of a virion. You have this cone, and inside of that cone is the viral genome. And I've been interested in this structure for since basically the mid 90s, and part of the inspiration for this line of investigation was to better understand that cone structure and how it worked in the viral life cycle, because there are so many events that have happened over time where people will see this and that, and it just became, everybody said, oh, it's uncoating. We don't understand it all, so, and I don't understand my stuff, so it has to be uncoating. And hopefully we're very close to ending that kind of a discussion. But anyway, so there were there's three basic models here. One is that when the cone you have fusion and the cone is dropped into the cytoplasm, that it just falls apart. We know that's not true today because things like TRIM5 alpha restriction, MXB restriction, these restriction factors interact with this cone. The other model is that the cone itself falls apart, and there's a lot of things that happen even into the nucleus that have determinants in capsid influence. So we recently reported, and don't have time to go into that today, but a little bit of the capsid could stay part of that all the way to integration, and that's what we've reported recently, and there's a consensus building on that. And then the other model is what we like to call cores at the pores. The idea that this core moves all the way to the nuclear pore, docks there, finishes up reverse transcribing and things, and then lets the genome go into the nucleus. And this is a favorite of many because of a variety of reasons. Some people would say, well, things like reverse transcriptase could float away if it wasn't inside of a bag. It could be possible. But we know there's a lot of these surveillance within the cytoplasm looking for things like DNA in the cytoplasm. There should not be DNA in the cytoplasm of a happy cell. And so that's something that's recognized, a pattern recognition of these PAMPs. It's recognized as being something foreign, and then it will set off the alarms, release interferon, and do all these other things. So the reason, what I would argue is that we know that the capsid itself can stimulate these restriction factors and signaling, and the nucleic acids can stimulate these pathways. So somewhere along the line, there's like a perfect time to switch. And I don't know when that is, and I don't think anybody in this room knows when that is. The virus knows exactly where that moment is to switch. Oops, what is, my computer's going crazy here. Okay, go away. All right, so one of the first ways that we labeled the virus and could follow it was by putting VPR in the virion. It interacts with GAG, and during assembly it gives us this green virions. And then some of the first work we did was to, when we started to look, the virus was moving in lines, and we were ultimately able to show that it was moving down microtubules, explained that movement. But what I want to talk about now is a completely different way to label the virion, and that's using a fluid phase marker. So you can, this was done by Ben Chen and other work by Greg Malikin. You put the GFP right into GAG between matrix and capsid with a protease site. And so as the virus assembles, it drags a bunch of GFP into the particle. But as the proteolysis happens, goes forth to make a mature virion, you basically release the GFP, but it's inside of the membrane of the virus, so it gets distributed. And I always argued that there was going to be some GFP in the cone, and there would be some GFP outside of the cone. Others felt it was all going to be on the outside of the cone. So we did some work including, say, you take the viruses and you lyse them and you run them out on a gradient. And so the cores fall apart, but you have some cores that can go into the gradient. And you can see the capsid here are these cores in the gradient, and then there's free GFP there, consistent with virus in the cores. And then, again, I don't have a lot of time to go into this, but TRIM5 alpha, if you interrupt proteasome function, will actually trap this cone intact, and it can't move forward. And when we do that kind of an experiment, we can show that a lot of these captured cones are full of GFP, so it's holding it intact. And in some work, I'm part of one of the Centers for Structural Biology, the Cheetah Group. They've been presenting actually just amazing cryo-EM pictures of TRIM sort of forming structures over this core consistent with holding it together. And that's that's what an artist said it would look like, and we can actually see it. It looks like that in those EMs. So we're going to try to leverage this capsid integrity, the ability to hold GFP in the core, and to follow this in a living cell. And so the idea is you have the fluid phase marker is spread throughout the virion. Upon fusion, you're going to lose most of that. The area of the cone is only about 20% of the total volume. So then you're going to go to less of the GFP, and then we have a cherry labeled VPR. And then when the cone starts to lose its integrity, you'll see the GFP will be leaking out. So, so that's what we can see in this movie, I hope. Yes, all right, good. So, so if you watch here, this is the particle, a lot of GFP. It just went down. That was the first fusion event, and then it just went away, even though the red color is still there. So that was the loss of the capsid integrity. I'll just do that one more time. So this is the quantification of that, and this is the particle within the field. So you have fusion, you lose a lot of the GFP, and then you here, that's the loss of the capsid integrity. It leaks out. So if we quantify that, what we find is that, we find a couple things. One is the science is never clean and nice. So we had reported using a different assay that the amount of time between fusion and uncoating was about 25 minutes. And so here with this assay, it was about 29 minutes, this one population. We saw two other populations. This one where it never held the GFP at all, and that's probably because of an aberrant core. You look at virions by EM, you can find cores with holes in them, cores that look like jelly rolls, all kinds of different structures. The one that was, if I might use the term annoying, was this species right here. And so this species never seemed to lose its GFP, right? So this could be the long intact core, the cores at the pores. So we needed some way to differentiate between these two, and we recently have achieved that. And so when I first started talking about looking at individual virions moving in cells, it was people said, well, Tom, how do you know that virion is the one that infected the cell? And and you keep asking me a question like that, I'll come up with lots of different answers over time. But my answer was, someday I'll be able to tell you that virus infected that cell. And so but when that day happens, you're going to have a problem, because when you show me a Western, I'm going to say, which protein in that band came from the virus that infected the cell? All of these other things where you're mashing stuff together, right? And we are at that day. So what we do is, basically what allows us to do this is a bit of brute force science and really good microscopes and other things. And so what we're going to do is use that 2-drop virus I showed you, and the genome of that, because it's all the gag is making GFP, if that infects a cell, it's going to turn the cell green. And then we have this ability to follow, and I'll show you this in a second, but we can follow multiple fields over time. And maybe I might not show you, actually. Anyway, we can follow multiple fields over time, and so we can look at 300 cells at a time. And so what we're doing is basically watching the cells for a day and a half. So we start in our early time window, taking image about every 3 minutes, a Z series throughout. You can't do that for a day and a half because you'll just cook the cells, but we can do that for a while, and then we can shift and have less. Have less. I don't know how this got out of order, but anyway, I'll go back. So here is just an example of how it works. So we can follow, and I'll just do this very briefly, but we can. You have over here a lot of different places. Each one of these is a place where the microscope is going to visit the cells and move back and forth from these. And then at each spot, it does a Z series in both colors. And you basically put in enough spots there so that it can look at all these cells, these fields of cells over time, and still be back to the first one after the 3-minute interval. And so, and if we do that then, we can do this kind of an experiment where we go, and then we also infect with basically 1 particle per cell, very few particles. The challenge there, this is actually a cell next to it, is that to get 1 particle per cell, then you have lots of cells that get no particles, et cetera. And so here is what's happening over the first 90 minutes. We see those 2 drops, and then now you just have to wait. But now you can see this cell is beginning to turn green. It's expressing the viral proteins. So that particle infected that cell, and it did that, okay? And you keep making the postdoc do it again and again and again and again. Basically, you map these, and there's some complexities because not every particle has the anchor, so you see it after the second color disappears, though you know, et cetera. Some cells have 2 particles happen to get in there, but every infectious event, when you go back in time, had a particle that had a second drop at this point, which is very consistent with what we see within that one population. So, and then there's a lot of other experiments, but I think those were CHO cells. It's kind of artificial, but we can do the same thing, not the infected cell yet, but we can do CD4 cells with wild type envelope. We can do monocyte-derived macrophages with VSVG, and we get this same sort of early uncoating step. So it seems to be the same in T cells, in macrophages, HeLa cells, CHO cells. The virus only knows one way to do it. And then one of the things we've showed is that if you slow reverse transcription, you slow that process. So even in the macrophage, you can delay this event by slowing reverse transcription and then washing out the nevirapine, and then it basically allows it to proceed. So the conclusions 3 is that uncoating takes place early with real viruses that infect real cells, about 30 minutes, and it is facilitated by reverse transcription. These general kinetics are independent of entry pathway and cell type. And then a little bit, and I didn't have time to show you this data, but a little bit of capsid remains with the structure to explain these later events that sort of drive these alternative models where, the capsid determinants in the capsid can influence the integration site, being a big example. So basically, we come back to our model. The data that I just presented to you is consistent with this idea that most of the capsid, also I didn't have time to go into it, but we've really extensively characterized this. So it's not like there's a hole there, but we see a fairly traumatic, catastrophic loss of capsid down to a small amount. So this seems to be the pathway that's associated with infection. So I'll go ahead and stop there and acknowledge people in my lab that have been involved with this work and other collaborators. And I have to admit, the last thing I put together was this acknowledgement slide, so I apologize because there could be some more names there. That was a lot of work over, some of it over a 15-year span that I presented there. So thank you very much, and I'll be glad to try to answer any questions.